by using design techniques that infiltrate, filter, store, and evaporate to detain runoff to its close.
Do you have any problem, Dr. Redillas?
Okay, I don't know.
Okay, Aimee, can you share screen the presentation, please? So that Dr. Redillas can continue.
Yeah, okay. Doc Alvin, I can.
Ray has been disconnected. Okay.
Yeah, okay. I can share the screen na lang. So I can control it.
Okay, sure.
Yeah.
Go ahead.
I don't know what happened. Ray is gone. Ayan, Ray entered again. I think there was a problem with his connection. Anyway, okay. Ray. Okay, he's there connecting. All right.
I'm
Okay.
sorry. I'm sorry for
Okay,
the...
go ahead, Ray. Please
Okay.
mind the time. Thank you.
Okay. So we are introducing now the low impact development. It's an innovative water approach. The main difference between the conventional and LID approach is that the conventional approach is that you just convey and discharge the water, while LID capture the water at its source, and it from the figure on the right side that it reduces the runoff of an area. The different types are the bioretention, rain garden, green roof, infiltration trench, permeable pavement, rain barrel, rooftop disconnection, and the USAPA. It was actually used around the globe in US, Korea, China, Australia, and UK with different names such as smart cities, WSUDs, and others. Okay, so next, the objectives. For the objectives, we have 4. First is to characterize the rainfall because different site has different rainfall intensity. So it is important to have a localized rainfall data. Next one is to determine the water balance of the site with and without the use of LID. The third one is to investigate the hydrologic performance of the LID with the varying percent imperviousness, varying subcatchments, and varying rainfall percentile. The last one, which is the main highlight of our study, is to assess the optimum green-gray ratio. Okay, so let us now go to the methodology of our study. So for the data gathering, 2 important data that we have should rainfall data and the digital terrain model. For the rainfall data, we have the 30 and 28 daily rainfall data from 1989 to 2018 of Ambolong, Batangas, and NAS UPLB. For the digital terrain model, it was gathered from the NAMRIA, or the National Mapping Resource Information Authority. So this was all used in the analyzing of the data. In our side of the actually it was changing. So from the pre-developed to a pre-developed area. Actually, the DLS Laguna is located in the southwestern part of Laguna Lake, and it is on the construction there is ongoing. So the next one is the processing of data. So the DTM, the digital terrain model from NAMBIA, was delineated using ArcMap, and we actually produced 17 subcatchments and chose the 3 subcatchments which main factors. For the rainfall data, we actually analyzed it since it is a daily rainfall data. The acceptable input in the stormwater management model is an hourly rainfall data. So we use a triangle method to disaggregate it. So since we do not have a localized data, we also use an inverse distance weighting method, and we use the Ambolong and the NAS UPLB to create a single rainfall data, and actually plotted it using Weibull and plotting position. Okay, so for the modeling and simulations, we actually use the stormwater management model. It is a hydrologic model to analyze the water quantity and water quality of an area. So these are the parameters of the subcatchments, and these are all the LED technologies that was used in SWMM: the bioretention cell, infiltration trench, rain garden, green roof, permeable pavement, rain barrel, rooftop disconnection, and vegetative swales. So these are all used in the simulations. Actually, there are a lot of LED, but these 8 are the only available in SWMM. Okay, so these are all the parameters. So these are all the scenarios and the notations. Also note that the SAC ratio used is 5% for the area of the LID facility to the area of the catchment area is only 5%. Okay. So next. So in total, for the water balance, we have a total of 135 scenarios, and the green-gray ratio as 675. So a total of 810 scenarios was conducted. So for the results and discussions, so these are the rainfall characteristics that was analyzed from the raw data from PAG-ASA. So as you can see, from January to April, we have low intensity of rainfall, and it increases from May to December. So when we design LID, it is important. It is good to design the LID based on the results from May to December. Also, on this part, the early time series of the rainfall depth of the 60, 70, up to 95th percentile rainfall data. So, needed yan to for our different scenarios. Okay. Also, the bottom part, the frequency distribution, as you can see, majority of the rainfall falls within less than 5 mm of the annual rainfall. So we do not need to design the LID. We do not need to design a bigger LID. So here on the right part, cumulative distribution function for the rainfall percentile, and we actually converted into the return period for us to analyze. Since in the Philippines, we are more on analyzing the data, more on return period. So the 30-year plan, 5-year plan, 10-year plan, and others. So for the water balance, I will show you one for the subcatchment one. So this is the water balance of the site with and without the use of LID with the following parameters of total infiltration, runoff, and storage. So among this LID, comparing to the scenario from the no LID, bioretention cell reduces more on the total runoff, infiltration trench, and for the total infiltration, and rain garden for the increase in storage. So same results happen to the subcatchment 2 and subcatchment 3. So same trend, and yan. And for the subcatchment 3. And to summarize this, next slide. So as we increase the subcatchment area, we actually decreasing the percent total runoff reduction and decrease the percent increase in total infiltration. So for the rainfall percentile, as we increase the rainfall percentile, we increase the percent of total runoff and decrease the total infiltration. So now we'll proceed to the LID hydrologic performance. So this is the overview of the simulations. Actually, we have 81 charts. Actually, we use the 3 subcatchments and the 9 LID with their specific traits for the total runoff, total infiltration, and storage. So this is the sample of the charts. And as you can see on this no LID part, that for all the LID used, you can see the decrease of the total runoff. So all from 60 to 98th percentile. And as we increase also the percent imperviousness of an area, we actually also increasing the total runoff of an area. Okay? So nakikita natin na the using LID, we actually decreasing the total runoff. For the total infiltration here, as you can see, for the actually you can see the effect of imperviousness, total infiltration in the infiltration trench. Actually, there is an increase from the no-LID to infiltration trench increase. Okay. So actually, the main idea here, the LID decreases the total runoff and increases the total infiltration. Also, for the storage, rain garden increases the total storage of the area. Yeah. So next, let's focus on the green-gray ratio of the water balance. So these are all the 8 LID with the no LID. So we will compare the effect of the green-gray ratio to the water balance elements, the runoff, storage, infiltration, and the infiltration storage. So these charts, as you can see, there is an intersection of the infiltration and storage and the total runoff. This intersection, there is a significant decrease in the total runoff and the increase in the infiltration plus storage. Okay, so that is significant, that the around 1:1 green-gray ratio. Also, please be note that the around 2.2 from all the graphs, that there is a slight changes that occurs. So actually dun tayo sa summary for the ano. So next. Okay. So actually same yung nangyari for all the LID. And most of the charts, actually 135 charts. And this is the summary. Actually, these are the results from the subcatchments 1, 2, and 3 of all the LIDs with the green-gray ratio from where they intersected. So 0.69, 0.99, and as you can see from the no LID, the bioretention cell decreases. We actually do not need a higher. So these are the green-gray ratio. Okay. Next, LID performance. Yan. Yan. As you can see here, for the optimum green-gray ratio, 2.2, nag-decrease yung sa runoff, increase in infiltration, and increase in storage in compared to the no LID. Yung dot, that is the no LID, and the chart are the for the different scenarios. Yung ano. Okay, so for the conclusions, I dictate. So design the LID based on the light test and heaviest rainfall that occurs in February and July. Designing a large LID is unnecessary because more frequent daily rainfall was less than 5 mm, which comprises 80% of the accumulated rainfall. LID can feasibly capture the 1-year return period of the LSU and let the conventional system address the remaining excess runoff to capture 25, 50, and 100 year. Bioretention cell and infiltration trench can be used effectively to reduce the total runoff by 20 to 90%. So infiltration trench and impermeable pavement can be used effectively to increase the total infiltration up to 42%. So smaller subcatchments produce greater reduction in runoff and greater increase in infiltration. So in this study, 68th percentile showed the most significant decrease in runoff and 95th percentile. So imperviousness is possibly correlated to total runoff and negatively correlated to infiltration. And last, the optimum green-gray ratio in the LSU Laguna is 11:5 or yung 2.2 and can be used in designing an LID after the calibration. Okay, so for the recommendation, use infiltration trench in the LSU Laguna campus as an initial study because it both increases infiltration and decreases runoff, better than the available LED technologies in SWM. Compare the effect of LED technologies on relatively smaller subcatchments and bigger subcatchments. Use the smallest possible subcatchments possible and use 80, 90, and 98% rainfall in the simulation. Quantify also the evapotranspiration, since in the study we did not include evapotranspiration. And last, calibrate it through actual and rainfall simulation. So that's all. Thank you.
Okay, thank you, Dr. Redillas and Mr. Frias, for that very interesting, study. We are not going to entertain any question at the moment. We will have to reserve that until the three other speakers for this part of the session. So in the meantime, you may write your questions on the chat box. Okay, I would like to remind again all our speakers to strictly observe the 15 minutes presentation. Okay, so let me now proceed with the next presentation on pharmaceutical residues and antibiotics in the coastal waters of the Philippines. A graduate student and researcher at the Marine Science Institute of the University of Diliman, Ms. Cheryl May F. Mariano. You can proceed now, Cheryl.
Okay, good day, everyone. So I will be presenting our study. And this study was conducted with Dr. Luisa Chilen Angeles and Dr. Diana Aga of University at Buffalo, and my mentor, Dr. Caroline Marie Haraula. So pharmaceuticals and personal care products, or PPCPs, are a group of products used by individuals for personal health or cosmetic reasons, or used by agribusiness to enhance growth or health of livestock based on the US EPA. And this includes but are not limited to antibiotics, fragrances, cosmetics, and hormones. The US EPA and the European Union considers PPCPs as emerging contaminants, which means that they are being detected in the environment at low concentrations. However, they have adverse effects. That's why they are being monitored by these government agencies. What are these adverse effects? So the most alarming of all is antibiotic resistance. And there are also noted long-term effects to ecological balance, negative growth effects, high probability of adverse effects through chronic exposure, and there are also noted biochemical effects on aquatic species. How do these PPCPs end up in the natural waters? So most of the sources actually come from terrestrial sources, from livestock farms, residential areas, industrial wastewaters, hospital wastewaters. When pharmaceuticals are taken in, especially antibiotics, not all of it is metabolized by the body, and a fraction of it is excreted as its original form or as a metabolite. And this is what ends up in the runoffs, in the wastewater, and then and ends up in the stream, sometimes in the groundwater, and ultimately in the coastal waters. For wastewater, usually it's being treated and it consequently removes PPCPs. However, it's not targeted, and some still persist and still end up in the coastal waters. Now we see that wastewaters are major carriers of PPCPs in the environment. How does the Philippines fare in wastewater management? Based on the government report, only 10% of wastewater in the Philippines is actually treated, and only 5% of the total population is connected to a sewer network. But of course, the government has tried to address this. There are several programs in the recent years trying to address this problem, one of which in Metro Manila is the Wastewater Management Project, spearheaded by Manila Water and Maynilad. So they have been constructing wastewater treatment plants around the region and also nearby provinces, which we can see in this figure. But then the rest of the country has still yet to catch up in this kind of development, but we are progressing in it. So for our method, so water quality in the Philippines is a major issue. So we want to know the current situation of pharmaceutical contamination in our coastal waters. So for our study, we sampled from different sites across the country. From Bolinao, Pangasinan, we have Manila Bay, Metro Manila, Mabini, Batangas, and Boracay Island, Aklan. We also sampled from the UNESCO World Heritage Site to Bataha Reef in Palawan. And then we sampled from Cagayan de Oro and Davao City. So these study sites were then grouped based on their major characteristics to aid in the interpretation of our data. Bolinao serves as our representative for fishery sites, and then Mabini, Batangas, and Boracay are tourist sites without major river waterways, in contrast with Cagayan de Oro and Davao, which are major river basins. And then we compare the assemblage of pharmaceuticals detected in a less impacted site to Bataha Reef to a heavily impacted site, Manila Bay. Our methodology follows BAT and AGA 2005, which was modified in 2018 by Angeles and AGA for freshwater and wastewater samples. Briefly, we collected duplicates of samples, processed it through filtration, acidification, injection of surrogate standard, and then we concentrated the target analytes through solid phase extraction, and then the eluent was then run through an LCMS instrument. For the identification and quantification, we looked at 37 pharmaceutical compounds, which includes common pharmaceuticals and 4 classes of antibiotics. For many samples, the standard wasn't detected, probably due to degradation because the time from analysis and sampling was a bit long. So we had to do a 1-point calibration instead. And since the method was optimized for freshwater and wastewater samples, we have to consider the effect of the ions in seawater, and thus we conducted a matrix matching for the seawater samples. So for the results, first let's look at Bolinao, Pangasinan. So we sampled from Gigiwanan Channel. So this is a fishery area. We have many fish pens, and we also sampled in the area of the giant clam nursery. For this site, we were only able to detect anhydroerythromycin. So this is a metabolite of erythromycin, a common macrolide. Erythromycin itself is an antibiotic that is cultured from soil bacteria. So these low concentrations, we're not exceeding 1.4 nanogram per liter, could be just an environmental background concentration, since we didn't detect other pharmaceuticals that indicate anthropogenic sources. And for the tourist sites, we have Mabini, Batangas, and Boracay Island, Aklan. So we know Boracay, but for Mabini is a known tourist dive site, and it's near the Verde Island Passage, which is a hotspot of marine biodiversity. So we are concerned what effect could the pharmaceutical contamination have with the biodiversity of the region. So we also looked into the pharmaceutical concentrations in this area. For Boracay, we had an extra sampling period during the period when it was closed from tourists, and then dry season and wet season sampling for both sites. First, let's look at Mabini, Batangas. So we sampled upstream and downstream in the drains. There is no major river in Mabini, Batangas. So these are just small waterways, small streams. And then we sampled along the coasts. Our upstream samples here on the right of the graph has low detections of caffeine and acetaminophen. Caffeine and acetaminophen have been used in many environmental studies as tracers of untreated wastewater. So their presence in the natural waters could indicate discharge of untreated wastewater somewhere. So upstream, where there are less people, less structures, we have little concentrations, but there's still some. But if we compare it to the downstream samples from the drainages and small streams near the residential areas, you have high concentrations of caffeine compared to the upstream sites. In the coastal sites, we're also able to detect caffeine and acetaminophen. So there is presence of untreated wastewater in the coastal waters of Mabini at less than 100 ppb for caffeine. Then let's look at Boracay. This sampling was when it was closed from tourists, but still there are residents in the area. Point of interest for this sampling are sites Inland 4 and Inland 8. Inland 4 is a mangrove site where the drainages from inland ultimately discharge into this site. And for Inland 8, this is a small wetland, an enclosed wetland surrounded by residential structures. And we see here caffeine concentrations were detected at around 400 ppb for the wetland and over 200 ppb for the drainage discharge. Acetaminophen was also detected for both of them. And then the offshore samples for this sampling period, the team tried to target suspected discharge sites of pipes offshore and also affected sites of drainage discharge. And we detected caffeine and acetaminophen in 4 of the offshore sites, offshore 3 and offshore 4, and offshore 6 and offshore 7. There are small detections in other offshore sites, though not as high as from the 3, 4, 6, 7 sites. Now we went back on April 2019. This was already a period where it's open to tourists. Inland 4 site, comparable to the previous sampling period, we have higher concentrations of caffeine. So again, Inland 4 site is the drainage discharge site. So it just reflects that there are more people that's discharging caffeine into the drains. Acetaminophen was also detected. This site, II, This part here is another drainage site that we found. It directly discharges into the sea. So and we detected caffeine at almost 200 ppb. Coastal sites, the ports here also had detections of caffeine below 50 ppb. And then for the September sampling, these are just offshore sites, and this is during the wet season. So rainfall could have affected the concentrations of the PPCPs detected. So all the concentrations were below 40 ppb, and most are caffeine. Acetaminophen was detected in one site in OE6. Okay, so this is OE6, this is near a pipe. Next, let's look at the river basins. So the river basins, we have major rivers draining into the sea. So we sampled along the river and then adjacent to the coastal waters. For Davao, we sampled during dry season and wet season. For Cagayan de Oro, we only sampled during 1 month in October 2019. Let's look first at the Davao results. In general, the upstream sites, we can see here in the right side of the presentation, we have here the upstream sites, Suwawan rivers until Lamanan River. And then we have the downstream sites here, Waan River, and then the Bangkerohan surface and bottom. In general, the upstream sites have lower concentrations of PPCPs detected compared to the downstream sites. And similar to Boracay and Mabini, we were only able to detect caffeine and acetaminophen. The solid colors represent the dry season sampling, and the hollow bars represent the wet season sampling. In general, we can also see that the dry season sampling seems to have a greater detections compared to the wet season sampling. And if we look at the seawater concentrations, this is the dry season and the wet season. We have, again, in general, higher concentrations during the dry season. And if we compare this concentration to the downstream sites, we can see that they're almost at the same range. It's also notable that the downstream sites is adjacent to the city center. So more structures, more people, and you have more discharge probably of untreated wastewater. The same trend can be seen in Cagayan de Oro. In this upper graph, we have here the coastal water samples, and below is the the river samples. And the range is just around 0 to 40 ppb for the coastal samples, 0 to 30 ppb, so it's just around the same range. Again, caffeine and acetaminophen were detected. We also sampled from another river stretch in this side, Agusan River, and we see caffeine detections at around the same range as well. So the similar range of the stream samples, the river samples, and the adjacent coastal water seems to suggest that the rivers are efficient conduits of pollutants to the coastal area. There is also another consideration, and that is for in the Davao area, the coast is also lined with other structures. It has residential areas, and the longshore current in this area uh, entrains the discharge from the coast and circulates it back to the river, to the outlet of the river. This is a sea surface circulation modeled by Villena in 2009 for April in Davao, which is this same as our sampling period. And we can see that if there are discharge here, it's being brought by the circulation back to the sampling site. So this could be contributing to our higher detections in the coastal areas compared to the streams. Another interesting find is that in Cagayan de Oro, we detected sulfametazine, an antibiotic used by both humans and animals for veterinary farms, which is used for growth of sheep, cattle, poultry, and pigs. And we even have higher concentrations near the coast. So we weren't able to identify the exact source, but this just suggests that upstream, there is source for these antibiotics, and probably it's an livestock farm. Lastly, we compared the assemblage of a less impacted site versus a heavily impacted site. In Tubataha Reef, we were only able to detect caffeine for most of the sites, and these are at low concentrations. And since there are no other caffeine sources around this area, this is most probably from anthropogenic sources, from the people that were staying in the area during the sampling period. Comparing it to Manila Bay, where we sampled in Pampanga River and Pasig River and other parts of Manila Bay, For Pampanga River, aside from acetaminophen and caffeine, we were able to detect anhydroerythromycin, similar to Bolinao. And for Pasig River and Manila Bay area, we detected even more pharmaceuticals, such as clarithromycin, acetylsulfamethoxazole, trimethoprim, carbamazepine, and diclofenac. So these are other types of antibiotics and pharmaceuticals. And these detections are actually comparable to what we can detect in wastewater treatment plants. So this graph on the left are detections from influents and effluents of a wastewater treatment plant. And in general, we can see that the concentration decreases once it's in the effluent, but pharmaceuticals still persist. And eventually, if this gets discharged, it's probably what we also detect in the coastal waters, aside from non-point sources. So in conclusion, caffeine and acetaminophen, PPCP is indicative of anthropogenic contamination, untreated wastewater specifically, are present in most to all coastal waters of the study sites, with concentrations reaching up to 230 ppb in Davao Gulf. Dilution and efficient transport of PPCPs contribute to detected concentrations, and sulfametazine, an antibiotic, is detected above its normal environmental concentration. The widespread detection of pharmaceuticals in the coastal waters indicate the extent of anthropogenic impact in the aquatic environment, and we have heavily impacted sites with the most assemblage of PPCPs. And lastly, stringent guidelines and early monitoring must be considered to prevent concentrations reaching sublethal to lethal levels, harmful to aquatic organisms and coastal communities. And that is all. Thank you.
Thank you, Cheryl. Again, please reserve your questions and type it in the chat box. We'll entertain them after two more presentations. Okay, again, this is your Rec 8. There are over 30 participants here. We may now proceed to the next presentation. The next presenter, Aimee, please. The next presenter will talk on the modeling of the fate of pharmaceutical pollutants in Manila Bay, Philippines, also a graduate student at the Marine Science Institute at UP Diliman. Ladies and gentlemen, Ms. Anne Elizabeth Rolyon Innova. Anne, please mind the time. It's on the screen by Blue Sky Timer. Thank you.
Okay. Good day. I'm Anne Elizabeth Rolyon Innova, and this is our study, Modeling the Fate of Pharmaceutical Pollutants in Manila Bay, Philippines, together with Diana Aga, Caroline Haraula, Shirel Mariano, and Dr. Cesar Villanoy. Pharmaceutical pollutants have various effects to aquatic life. They may promote antibiotic resistance. They may be toxic to bacteria in sewerage treatment facilities. Pharmaceutical products come from factories and they go to our residential houses, hospitals, and some of them are being treated in wastewater treatment plants. However, not all of them are removed in these wastewater treatment plants, and most of them go directly to our rivers and oceans through the sewage and through our urine. Another pathway is through agriculture and livestock, which find their way into our rivers and oceans through runoff. Our study area is in Manila Bay. Why Manila Bay? It is surrounded by our biggest and highly populated provinces. It is surrounded by a lot of hospitals. All the pins here are hospitals and residential houses, and also, on the other hand, agricultural and aquaculture areas. Manila Bay has been previously studied for pharmaceutical concentration by Mariano, Singh et al. And sampled in 2016, and they found that in Pampanga River, there were 5 nanograms per liter of total pharmaceuticals, while in Pasig River, there were 76 nanograms per liter. However, we do not know the concentration inside Manila Bay. Manila Bay has been previously modeled by De Las Alas and Sudusa 1985 and Villanoy and Martin 1997. However, these models are mostly wind and tide driven only. Rivers were not accounted in these models, especially when Manila Bay is an estuary and the rivers contribute to a large volumetric flow of Manila Bay. Thus, we have the following gaps. What is the resulting water circulation inside Manila Bay due to tides, winds, and rivers? And where are the pharmaceutical pollutants being concentrated? And with that, we have the following objectives: to create a tidal model of Manila Bay with effects of tides, winds, and rivers, and to determine areas of high pollutant concentration. How did we do this? We started with our coastline, land boundary, and bathymetry, and from that, we made an overall grid. We gathered more information such as tides, winds, salinity, rivers, pollutants, and inputted that to the Delft 3D flow model. From there, we get our output, which is the velocity and pollutant concentration. Manila Bay has a depth of 0 to 70 meters with mostly 30 meters in the inside of the bay, and the shallowest areas are in Pampanga, Bulacan, Manila with only less than 20 meters of depth. We used 15 layers with the thickness of the layer as a percentage of depth. We used 2 points at the mouth of the bay to define our open boundary, and that is in Mariveles, Bataan, and in Ternate, Cavite. 1 month of tidal data was used, that is from March and September 2021. And from that, we computed our major tidal constants using MATLAB T-tide. We inputted 1 month of wind data, and that is March, northeast winds, and September, southwest winds. The wind velocity is mostly 1 to 5 meters per second, and we applied a moving mean or average to smoothen the wind data. For the rivers, we accounted 6 major rivers: Pampanga, Bulacan, Pasig, Parañaque, Imus, Balanga. And we used the flow from the annual mean volumetric flow of the rivers from Alejandrino, 1976. And for the total pharmaceutical pollutant concentration, we used the data from Mariano Singh et al. In 2016. We also used the individual concentrations of anhydroerythromycin and caffeine. Now, let's move on to the results of our model. First, let's look at the mean velocity during March north winds. The left figure is the magnitude of the velocity, while the right figure shows the vectors or the direction. Water goes in from the Ternate Cavite area to the inside of the bay and goes out from Pampanga going to Bataan coastline. There are many counter-rotating jars on the inside north middle part of the bay, and these jars may promote the accumulation of micropollutants on the center of the jars. During south winds, September 2021, the trend is generally the same. The water goes in from Cavite, goes inside the bay, and goes from Pampanga to the Bataan coastline. The difference here is the flow from Pampanga is going to Pampanga, Bulacan, then Manila, and that the 2 counter-rotating jars are more prominent and stronger. Let's compare our model with the previous wind-driven model by De Las Alas and Sudusta. For our March model, north winds, the major difference is in Pasig River, in which the wind model says that the flow should be going to the northeast, but our model shows that the flow is going from Pasig River to the inside of the bay. This is because of the volumetric flow of Pasig River pushing the water there. For the September model, the major difference is in Pampanga, in which the wind-driven model says that it should be going up to Pampanga, while our model shows that the water is going from Pampanga River going south. And this is because of the contribution of the large volumetric flow rate of Pampanga River pushing the water southwards towards going out of the bay. Let's look at the total pharmaceutical concentration as a result of the water circulation. These graphs are, the maximum is at 8 nanograms per liter, and we can see that the highest concentration is at Manila from Pasig River going to Bulacan and then to Pampanga during the March winds. But in September, the concentration is from Manila going to Bulacan and going to Cavite southwards. Let's look at another set of figures, and this shows the contours and shows a 10 nanogram per liter maximum. And let us note that the European Union has set 10 nanogram per liter as their action limit. saying that beyond 10 nanograms per liter, they would require a special risk assessment for the pharmaceutical product. So here, the areas in dark red shows a 10 nanogram per liter and more of pharmaceutical pollutant concentration inside Manila Bay. Let's look at anhydroerythromycin. We can see here that in Pampanga, anhydroerythromycin is more red. This is because anhydroerythromycin in Pampanga River and in Pasig River has a more similar or more closer concentration with a 1:12 ratio compared to the total pharmaceuticals. Now, we might wonder why there is more anhydroerythromycin in Pampanga, knowing that it is surrounded by the agricultural area. So erythromycin is also used in agriculture. That is possibly why we also detect anhydroerythromycin here in Pampanga River area. Now we look at caffeine. For caffeine, there is almost 0 nanogram per liter of concentration in the Pampanga River. And that is because our residential houses in Manila, it is very close to the coastline. But in Pampanga, this area is mostly agricultural and our houses are further north of Manila Bay. We can also notice here in the model that in March, caffeine is going from Pasig to Bulacan and it crosses to Pampanga. But here in September, caffeine goes from Pasig to Bulacan and goes south, but it doesn't cross here. Here, it doesn't cross because of the 2 counter-rotating gyres and the reverse flow that may be preventing the pollutant from going to the other part or to Pampanga part of Manila Bay. Let's look at the vertical profile of the pollutant going from South Corregidor to North Pampanga, and it would look like this. River freshwater is on top while salty water is at the bottom. They don't easily mix because they have different densities due to their salinity. And because of this, our pollutants which are dissolved in the freshwater also stay on top. The problem with this is that our phytoplankton and photosynthetic bacteria also are on the upper part of the water column. Thus, they may be the ones greatly affected by the high pollutant concentration. Let's look at another vertical profile coming from Manila to Pampanga, and that looks like this. Now, this profile is shallower. It's only up to 10 meters, and we can notice here that more pollutants reach the middle or the bottom, especially in Pampanga area, it reaches the bottom. So the problem with this is the fishes and aquaculture areas are mostly here in the shallow areas. So they may be the ones greatly affected with the high pollutant concentration. We also look at another spatial concentration of pollutants from the top layer to the bottom layer. And we can see that the high pollutant concentration is at Pampanga, Bulacan area. And again, that are the shallow areas near the agricultural and aquaculture areas. Thus, they may be affected by this pollutant concentration. So in conclusion, we have created a 3D model of Manila Bay. And we know now that the wind and river flows have great influence on water circulation and in the dispersal of pollutants. High pollutant concentration are observed in Pasig River to the center of the bay, and going to Bulacan area during the north winds, and going to the Cavite area during the south winds. Dissolved pollutants stay on the top layers. For our recommendations, control and monitoring of pollutants coming from the source should be done. We should monitor our water quality of the river discharge and other point sources. We should review our current effluent guidelines, and we should have proper disposal or return methods of pharmaceuticals, more wastewater treatment plants, and technological improvement for the treatment of pharmaceuticals such as tertiary treatment plants. And for further studies, this model can be further improved with more runs with more data from other months. And this model can be used as a basis of sampling design considering areas of high concentration and prevailing predicted dispersal patterns. Furthermore, More investigation on the effects of pharmaceuticals on aquatic life should be done. Here are our references for the studies, methods, and data sources. And we would like to thank the Marine Science Institute, DOST, Oasis Laboratory, University at Buffalo, Randolph Singh, and the softwares, Deltares, and MATLAB. And with this, we would like to invite you to further participate in the research on the different aspects of pharmaceuticals in our country or in the aquatic life. Thank you very much and have a great day.
Thank you very much, Elise. Yes, indeed, it would be an interesting later on how the work of Cheryl also relate with your work here. Anyway, thank you. And again, reserve your questions until we finish with our fourth presenter in this particular part of the session. Okay, Amy. All right. So the fourth presenter in this part would speak on the extremophile diversity and functional annotation of bacteria as descriptors of hydrothermal geochemistry in hot submarine groundwater discharge in Mabini, Batangas, a research microbiologist also at the Marine Science Institute at UP Diliman. Let us welcome Aljon Francis Koji Elegado. Aljon.
Thank you, sir, for the introduction. Would like to share my screen. Let me try to, yeah, there we go. Okay, good morning, everyone from the Philippines, and good evening to those in the US. I am very grateful to be here and to be able to share my presentation. To properly introduce our research, I would like to first give a background on marine microbes and biodiversity. In this aspect, the Philippines is at a great advantage. Did you know that we are at the center of marine biodiversity? And we owe this biodiversity to biological productivity. Productivity, or as we often describe as how much marine life can be supported in a given space and time, is primarily supported by photosynthetic microbes, or in more familiar terms, plankton. So much so that plankton are a good source or a good measure of productivity in marine ecosystems. Plankton are at the base of marine food chains, and we owe biodiversity to these tiny organisms. Our key research interest is to look into these microscopic life forms and to go deep beyond what light can reach. You have your phytoplankton that needs sunlight, and there's also other microbes that are more versatile. Some thrive in the dark depths of the sea, right within hydrothermal vents, surviving above boiling temperatures and very strong acids. These extreme life forms have interesting relationships with the geological setting present in hydrothermal vents. They are so intertwined that we look very close at the geochemistry along with microbiology. So now, why do we study them? Because even from these processes that are almost alien to us, we still reap benefits. These microbial life in hydrothermal vents has a potential global influence, and these deep sea hydrothermal chimneys can be important sources of trace nutrients. That's why both microbes and geochemistry from hydrothermal vents are being studied in global research cruises. Hence, you have your geotraces and entire ocean expeditions. In addition, hydrothermal systems in the natural world are often hotspots for new discoveries. For example, the discovery and study of the Grand Prismatic Hot Springs in Yellowstone National Park, and later on the discovery of Lost City alkaline hydrothermal vents pushed the boundaries of life as we know it. And we also thank microbes for these discoveries, extremophiles that survive in such boundary conditions. And we attribute these discoveries in the exciting combination of geochemistry and microbiology. Shallow sea hydrothermal vents are relatively new in the playing field. Shallow vents were previously neglected, unlike their more popular cousins, the deep sea hydrothermal vents. But now, they're also recently getting a share in the spotlight in science. One unique trait of shallow vents is that they have a lot of gas caused by the low atmospheric pressure. And in addition, there's the gas exsolution or degassing that often leads to highly acidic fluids and enhanced leaching of metals such as iron or arsenic. So these characteristics are true for our selected study site, a 15-meter deep, carbon dioxide-rich, shallow hydrothermal vent situated in Mabini, Batangas, Philippines. We've recorded acidic fluid with temperatures as high as 85 degrees Celsius. There's also a lot of iron in this vent and not so much oxygen. So we would like to share some of our results to you in this presentation entitled Extremophile Diversity and Functional Annotation of Bacteria as Descriptors of Hydrothermal Geochemistry in Hot Submarine Groundwater Discharge in Mabini, Batangas, Philippines. We look at the microbes as important contributors to productivity and diversity in coral reefs and have a closer look at their potential function and extremophilic members to give a context of the hydrothermal biogeochemistry in the site located at the Center of Marine Biodiversity. So we first provide the setting, an overview of the environment or the niche that we expect our extremophiles to thrive in. So there's high temperature fluids, low oxygen, a lot of iron, up to 800 parts per billion, thanks to its acidity, pH of 5.88. The sediment biomass is potentially unique too. This plot on the right shows differentiated reservoirs of organic carbon. Sediments from the vent displayed in orange are unique from the background displayed in gray. Mats, oftentimes an example of accumulation of microbial biomass, were also discovered right next to the hot fluids. And these mats, they also have a unique bulk organic carbon signature shown in red. So unique settings lead to unique biology, and here of the extreme type. We found a lot of aquificae in our 4 mat samples. These 4 columns denoted as M1 to M4, and these aquificae are bacteria often described in terrestrial hot springs. And there's also a lot of thermophiles in our vent samples, including those from the vent sediments. There's also some high heat loving archaea. You have your Archaea globus and Thermococcus under the order Archaea globalis and Thermococcales. And there's order Hydrothermarchaeota, which was first described in deep sea hydrothermal environments. So that's quite unique, considering that these are shallow settings. So they're right next to our thriving reefs in Mabini, Batangas, with corals and seagrass meadows. So now that we know what microbes are present, what do these microbes do? So we tried to take a peek by annotating their known function in databases. So that we were able to do that thanks to this tool called FAPROTAX, or Functional Annotation of Prokaryotic Taxa. So we see some very interesting stuff here. We have methylotrophy, methanotrophy. So there must be, or there could be some methane in there that we have yet to measure. And there's also the null gas bacteria, those that eat or use up hydrogen for their energy. And we see that these groups that we were able to find correlate with vent geochemistry, the high iron, high arsenic, depleted organic carbon, and high organic carbon. So what else did we find? Remember that we found lots of iron in our vent. We were also able to find bacteria that use of iron as electron acceptor. So both in some of the mat samples and also in our vent sediments. So these iron bacteria were recently classified and described by microbiologists as zeta proteobacteria. And they have some very interesting biology. As context, iron is a trace element in seawater. And there's growing evidence that hydrothermal vents and microbes have a role in distributing iron in seawater globally. So back to our data, in pure cultures, the zeta proteobacterial mats are mostly iron oxides and not really biomass. So here in the bottom figure, which is from a study that isolated zeta proteobacteria, there's quite a small bacterial biomass to iron oxide ratio in terms of mat components. And similarly, in our samples, when we looked at them through confocal microscopy, There's also not a lot of bacteria or part of the mat matrix can be stained, showing relatively small amounts of bacterial biomass. So we have reason to believe that our mats are formed by zeta proteobacteria. We wanted to have a further look in the iron oxidizers in our samples, and thankfully there's a tool to compare our zetas to those in other studies. This tool is called Zeta Hunter. And first, we found out there's a lot of them in our vent sediments, and not just one genus of Zeta proteobacteria. And that some of the Zeta OTUs that we found have not been described in literature yet. So we have some novel Zeta OTUs that we were able to describe. Another interesting thing we found is that in sediments, the presence or their presence or their abundance coincide well with our geochemical measurements. Lots of iron in sediments coincided with high zetas or Zeta proteobacteria. Some appeared to be more influenced than the others. So here, we're also excited to see what other factors influence their abundance. So that's something that we're planning to do later on. So, so that's where our results end so far. As a teaser of what's to come, we are also looking into lipid data. Here we have viable cell fatty acid data, and this gives us more clues in their biology and ecological roles, and what compounds do they potentially leave behind to tell us of their activity. And that can be done not only in the present time, but we can also look at even the past, provided that these compounds are preserved in our matrix. Okay, so that's all. This research would not be possible without the help of the following individuals and organizations. We thank Planet Time Resort of Mabini, Batangas for their assistance. We also thank Jason Balaibo, Jika Domingo for joining us in sample and data collection via scuba diving. And we thank Thermo Fisher Scientific and Brownstone Asia Tech for technical and logistic assistance. This research was supported by the Department of Science and Technology, University of the Philippines, and Chad Picard. Thank you very much, everyone, for listening.
Thank you, Aljon. That's also a very good, study. And now we are ready for the question and answer. And if there's anyone on the floor who may want to ask, you may want to do that. Otherwise, I would like to request, any question on the floor. Perhaps Cynthia, is there any question there?
We do not have any questions on chat nor on YouTube, unless anyone on our participants, all of the participants, do you any questions? Suppose none. So my question is for Doc Marla Redillas. What are the policy implications of your findings, if you shared it with local politicians, local decision makers?
Thank you, Cynthia, for your question. Actually, for the study of low impact development, the main policy is for the control of non-point sources. So based on the other speakers, they mentioned about runoff, they mentioned about like in Manila Bay. So we can control and possibly treat those runoff that's going to enter in our surface water bodies. So there are other pollutants that can be carried off by stormwater runoff, especially in developed areas such as transportation land use areas, also in agriculture. So by applying this LID, we could possibly develop guidelines or measures, best management practices in order to also protect our water quality.
Thank you, Dr. Redillas. Aimee, a question from your end?
Okay, there's a question in the chat box. Okay, for Aljon, do iron oxides precipitate in the water column as the vent water mixes with the ocean water?
Yes. Oh, yes. That's one of the phenomena that happens when you have dissolved and reduced iron that's present in the vent water. when it mixes with the ocean water, which is highly oxidized, these precipitate. So that's one reason why there's also a lot of iron in the sediments that we found. For in case of the mats, there's possibility that the microbes are also enhancing or facilitating the precipitation of these iron. So I think, yeah, I hope that answers the question.
Thank you, Aljon. Question for the research on pharmaceutical. Cynthia, anything there?
No questions on pharmaceuticals. I guess I have a question on the tidal model for Elise. A great presentation, everyone. But Elise, what's the resolution of that model?
I think per grid, it has different resolution, but per grid in that model, it's by 0.5 kilometers.
Ah, got it. All
We
right.
can make a greater resolution if we need to.
That's
Thank
great.
you.
Yeah. What are your plans with the data that you created?
My plans. So I now have the overall model. So my further plans are to make nested models of the coastal areas such as in Pampanga Bay and in the Pasig area. I also am trying to make nested models with a higher resolution in that area. And another plan for that model is to include the sediment. So right now we have modeled the dissolved pollutants. So the next step for me is also to check if there are sediment in the coming from the river and their trends. Yeah,
Thank
that's
you,
my plan.
Elise.
Thank you so
Thank
much.
you. Amy, any question on the other speaker?
There are no more questions, sir.
None? Okay. So again, thank you so much, Dr. Redilias and Ray, Cheryl, and Aljon. Keep up the good work, excellent work. You know, there will be many opportunities in PAASE for us to update our work, and we look forward to your active, engagement with us. So again, thank you so much as we continue to the next part of our presentations. We have a faculty of the chemical department from Eastern Visayas State University, currently doing his PhD in chemical engineering at De La Salle University, to speak on the synergistic effect of the carbon dioxide ethanol water system on fatty acid ethyl ester production using wet feedstock. Ladies and gentlemen, let's welcome Jovenil E. Cartel. Amy.
Trying to play it, sir.
Hear any sound?
Methodology, research.
Good.
Hello everyone. Please allow me to present our research study entitled In situ fatty acid ethyl ester production from wet mahogany seed using supercritical carbon dioxide and ethanol. This presentation will include introduction, objectives, methodology, results, the summary findings, and the conclusion. So let's start with the background of the study. Biodiesel is an ester product composed of a mixture of alkyl esters. It is a renewable fuel that could serve as an alternative for fossil fuels. The major challenge in the widespread use of biodiesel lies primarily in the cost and the current technology for processing a wide range of feedstocks. In some ways, to reduce the cost in producing biodiesel and dependence on edible oil is to use non-edible vegetable oils, using cheap reagents, designing simpler and shorter process steps, which may support the UN SDG and 12. So if these challenges can be resolved, more opportunities will come in, like providing a biodiesel that offers competitive price with fossil diesel in the market. Next, let's take a look on the developments in biodiesel production through the illustration shown on the screen. Various processes have been developed for the production of biodiesel. An obvious difference in the major production step is the need to extract and the possible need to refine the lipid material from solid feedstock in separate processing steps. Eliminating these steps not only reduces the production steps but may also result in a lower initial investment cost and reduced overhead cost. Further lowering of the initial investment cost and reduction of overhead cost could be possibly achieved by eliminating the pretreatment steps, which is proposed in this study. In effect of the introduction of isotropic ethanol and wet feedstock, trans-esterification and esterification, that is with prior hydrolysis, may occur simultaneously in the process. This illustration will give an idea on how we have able to theorize our research study. And based on our hypothesis, the supercritical carbon dioxide will provide an extrusion and swelling strength against the seed matrix, which significantly could enhance the diffusion of the bulk solvent, which is the ethanol, through the solute-containing seed matrix. Since the bulk solvent is aqueous, the presence of water could promote hydrolysis reaction on the cell wall that hastens the extraction process. Furthermore, the reaction of the carbon dioxide and water present in the reaction mixture can result in carbonic acid formation or dissociation. Subsequently, the presence of this catalyst could serve as a cheap in situ catalyst in the reaction. And previous study also suggests that ethanol may also act as an acid catalyst at supercritical conditions. Thus, with the enhanced excretion and swelling strength, formation of in situ catalysts increased miscibility of the solvent in oil, enhanced diffusion of the bulk solvent through the solute-containing seed matrix. It is thereby possible to obtain an increased extraction efficiency, conversion, and yield. This study aims to provide comparison between the conventional and the proposed method and investigate on the synergistic effect of the supercritical carbon dioxide, ethanol, water system in the production of fatty acid ethyl ester using wet feedstock. Procedure started with the pretreatment steps, which include removing of seed pods from the seed milling from the seeds, and then the milling, and then the sieving. And experimental runs then proceeded using ethanol as reagent with the aid of supercritical carbon dioxide. Different treatments and process conditions were set according to the design of experiments, and all runs were done in duplicates with control, that is without the solvent. And then after the run, crude sample goes to the downstream processing, which includes centrifugation, the pH measurement, water scavenging, and filtration processes. The purified samples were analyzed in HPLC-LSD for fatty acid ethyl ester analysis, and prior fatty acid profiling was done via GC analysis. On the other hand, the spent biomass was recovered and analyzed gravimetrically to quantify the residual oil and eventually estimate the amount of oil recovered in the process. Morphological analysis was also done using the fresh spent biomass. The degree of conversion was compared in different methods considered in this study, which include the conventional technique, the in situ process similar to previous studies, which is the treatment 1, and the proposed novel technique, which is the treatment 2.
So good day, everyone.
The conventional technique may have higher conversion than the proposed novel technique. However, this can be well compensated with the cheaper cost of solvent and less environmental risk. Interestingly, the synergistic effect of carbon dioxide water ethanol solvent system had contributed dramatic increase compared when using only one or two solvents. Higher water loading in the proposed novel technique showed to be an advantage. The increase in volume expansion coefficient, or V over VO, of the solvents resulted in an increase in the oil recovery by 11%. In this effect, about 6% increase in yield was also achieved. The highest oil recovery and yield was obtained at conditions where the volume expansion coefficient was also highest at 2.065 and the pH was lowest at 3.71. Based on experiments, an increase of oil recovery was recorded at the higher volume expansion of 1.896 when using treatment 2 as compared to treatment 1. Further increase in the amount of hydronium ion via pH lowering from 3.8 to 3.71 and the increase of water loading from 0.7% by weight to 6.19% by weight lead to a higher recovery of oil and yield. The decrease of the conversion by 3% and the yield, despite the increase of the oil recovery, an increased volume expansion could be attributed to some phenomenal effects during phase equilibrium, chemical equilibrium, or its kinetics, either individually or collectively. The increased amount of water in the feed dilutes the concentration of reactant so that the kinetic effects can occur. Besides, the water in the field will reduce the equilibrium conversion and influence phase behavior. So chemical equilibrium and phase equilibrium effects could occur respectively. Furthermore, when more water is added, the vapor portion increases and the water in the liquid phase reduces, which promotes enhanced volume expansion. But unfortunately, this does not warrant an increase of conversion nor the yield. It is known that the oil is present in the cytoplasm of the cotyledon cells, alluronic cells, and radicle cells of the seed. Hence, the destruction of these parts of the cells is crucial to reactive extraction. So, using morphological analysis, which we could see how the seed is destructed to remove the oils. The structure of the fresh sample shown in figure A was composed of compact objects with smooth cavities and compact cells scattered after extraction, and sharp cavities were observed in figures B and C, suggesting the disruption of the cell wall. The difference of morphology of the spent biomass between treatment 2 and treatment 1 are quite distinct. Treatment 2 has more fine flakes, indicating an enhanced interaction of bulk solvent to the seed matrix, thereby increasing the recovery. Eliminating pre-treatment steps and utilizing isotropic ethanol in supercritical carbon dioxide reaction extraction was found possible. It was found out that the higher water loading of the proposed novel technique showed to be an advantage. The increase in volume expansion coefficient of the solvent resulted in an increase in recovery and yield. Further increase in the amount of hydronium ion via pH lowering led to a higher recovery and yield. These results were supported with the morphological analysis of biomass. With all these significant findings, it can be concluded that the synergy of in situ formation of carbonic acid and supercritical carbon dioxide expanded ethanol water solvent system has a relative positive effect on the production of fatty acid ethyl ester that introduces a novel technique that can be more economical with low environmental impact and sustainable. However, further studies is recommended to improve the degree of conversion and yield.
Okay. So thank you,
This research
Engineer.
study was supported by the Japan Student Services Organization and this work was also partially supported by eAsia project on bioenergy. This study could not be made possible without the support of my advisor in the Philippines, Professor Dr. Oresenia from De La Salle University, and of course to my mentors in Kumamoto University, Professor Dr. Armando Kitain and Professor Dr. Tetsuya Kida. And of course to my ever supportive mentor, Dr. Alvin Kulaba, and my employer, Eastern Visayas State University. Thank you so much for your attention. I also would like to thank Paase and everybody involved in organizing this forum. It is indeed an honor and a privilege to share our research output in this technical session. God bless us all.
Thank you, Engineer Cartel, for that wonderful also work, especially with your collaborators in Japan. We now proceed with our next presenter. Okay, Aimee, can you show the bio note, please? Right. Again, our next presenter is an instructor at the Department of Chemical Engineering at UP Diliman. His research interest is in multi-physics modeling and energy systems modeling. To present from idealized battery models to equivalent circuit models using multi-physics and reduced order modeling, may we have the pleasure of introducing Mr. Michael Castro. Michael, your floor.
I'll be discussing my research entitled Development of Reduced Order Equivalent Circuit Models for the Energy System Modeling of Lithium-ion Batteries. So this presentation is divided into 4 parts. First, I'll introduce lithium-ion batteries, why they're important, and how they're being modeled. Second, I'll discuss the objectives and the methodology of the study. Third, I'll go over the results and discussion. And finally, I'll give the conclusions. Okay. So the world is undergoing a transition to renewable energy. It is also happening in the Philippines, especially in off-grid areas, due to the rapidly declining costs of renewable energy technologies and the potential of solar and wind in the country. At the heart of every renewable energy installation is an energy storage technology, which stabilizes the power output of renewable energy technologies. So lithium-ion batteries are ideal for this purpose because they come in a variety of chemistries with various advantages. For instance, the lithium iron phosphate or LFP chemistry offers low cost and thermal stability, whereas the nickel cobalt aluminum oxide or NCA, as well as the nickel manganese cobalt or NMC chemistries offer high energy density. Now, the deployment of lithium-ion batteries in renewable energy systems is studied using energy systems modeling software, such as Homer Pro, which is the leading commercial energy systems modeling tool. So this software has 2 important tasks. First is to simulate the battery by keeping track of its SOC or state of charge. The SOC is defined as the charge in the battery divided by the maximum charge that you can store. Second is to optimize the battery by determining its energy rating or its size that minimizes the cost of generating electricity. So to do this, the energy models will typically use an idealized battery model. So here we model the battery as the tank of charge, whereas energy losses are captured by efficiency parameters. However, this is a very simple description of the battery. Now, there are other more complex models. However, these require experimental data, and these are also developed for lead-acid batteries. So it's unclear if they're applicable to lithium-ion batteries. Now, on the other hand, we also have multi-physics models. So here we simulate a battery using fundamental laws in physics and chemistry. So this is mathematically rigorous. And also, this requires less experiments because the data that you need is already published in literature. These are also chemistry specific. So it can account for the differences between cell chemistries. Now, when you model a lithium-ion battery, we typically use the pseudo-2D or the P2D model, which couples 1D transport across the battery and 1D diffusion into the electrode particles. However, it is too complex for energy systems modeling. So to bridge the gap between energy systems and multi-physics models, we use reduced order models. So in reduced order model, we take a multi-phase model and simplify it while preserving its accuracy or predictive power. So there are many ways to do this. However, we'll be using the reduced order
Parallels.
equivalence circuit models. So in an equivalence circuit model or ECM, we model the battery as a voltage element, whereas energy losses are captured by impedances such as resistors and capacitors. So these are simple enough to be used in energy modeling, and they also preserve the cell chemistry because they come from multiphysics models. So in this work, we want to determine if the idealized battery model is in fact a viable or a appropriate method of modeling a lithium-ion battery by comparing it with reduced order ECMs for lithium-ion LFP, NCA, and NMC. So to do this, we will use 4 steps. So first is to replicate the multi-physics models of lithium-ion LFP, NCA, and NMC batteries reported in literature using the COMSOL multi-physics software. We'll then take the multi-physics models and reduce them into appropriate ECMs for each chemistry. These ECMs will then be implemented into ISLA, or the Ion Systems LCOE minimization algorithm, which is our in-house energy systems modeling tool. And finally, we'll compare the idealized battery model and the reduced order ECMs in terms of their SOC prediction and optimum sizes. So for the multi-physics models, we will be using the following studies because their authors gave a complete set of data which you can use to verify the model and build the multi-physics model. So we'll couple the pseudo to the electrochemical model, which describes one layer of current collectors, electrodes, and a separator, and the 3D thermal model, which describes the entire battery. So once we have our multi-phase models, we can then reduce them into equivalent circuit models. So we'll do that in 3 steps. First is to estimate the open circuit voltage or V naught of the battery based on material properties, which will also input to the multi-physics model. We'll then estimate the impedances, the resistances, and the capacitances using the hybrid pulse power characterization or HPPC test, which I'll discuss in the next slide. And third, we'll select the best ECM in terms of its accuracy of voltage prediction versus the multi-physics model. Okay. So about the HPPC, or about the HPPC rather. So first we'll input the current pulse to the multi-physics model. We'll then get this corresponding voltage response. Okay. So to estimate the series resistance or R naught, we'll base that on the initial jump of voltage or the initial voltage jump upon the application of current. We'll then compute the RC time constant tau sub n by fitting the voltage response after applying currents to the point D to point A by fitting that curve into this equation. Finally, we'll estimate the RC resistance R sub n by fitting the voltage response after applying or during the application of current from point B to point C. By fitting this curve into this equation, we should get our RC resistance. Okay. So we'll estimate this at 30 degrees Celsius and 50% SOC. So once we have our reduced order ECMs, we can then implement that into ISLA. So we'll need to use 2 correlations to do this. First is the power versus current relation shown here, which converts the power used by ISLA to the current which is used by the reduced order ECM. This also will help us convert the restrictions on the current into restrictions on the power. Next, once we know the current, we can update the SOC, as you can see here. Now, to further simplify this equation, we'll assume that the V naught or the open circuit voltage is the piecewise linear function of the charged capacity or Q naught or QT. Okay. And then to compare the idealized battery model and reduced order ECMs, we'll consider a variety of case studies. In particular, we'll consider a large island called Sibuyan Island, a small island called Lapidigan Island, and a household. So for the off-grid islands, or for these 2 islands, we'll consider 3 scenarios. So solar battery diesel, and solar wind battery diesel. As for the household, we'll consider two scenarios, that's solar battery and solar battery with grid connection. Now here we'll compare the batteries using a simulation case study and optimization case study. So under the simulation, we will just simulate the systems. We'll compare the SOC predicted by the idealized battery model and the reduced order ACMs. And as for the optimization, we'll determine the sizes or the energy ratings of the batteries that minimize the energy costs. Okay. Now for our results. So here you have your comparison between the multi-physics models and the experimental data for the voltage and temperature. So you can see that there's generally good agreement. So this is for LFP, but you can observe the same thing for NCA as well as the NMC chemistry. So next we have here the reduced order models. So again, we have 3 reduced order models for each chemistry, or rather 3 ECMs for each chemistry, but we only want to select one. So we'll choose the first order RC because we determined that after doing a pulse test and a full discharge, the first order RC model has the most accurate prediction compared versus the multi-physics model. So we'll select this for implementation. Next, we have here a comparison between the SOCs predicted by the reduced order model and the idealized battery model. So we found here that this is a direct comparison of the SOCs. And on the right, you have the SOC predicted by the reduced order ECMs minus that predicted by the idealized battery model. So we find here the LFP battery has the closest agreement with the idealized battery model, whereas the NCA and NMC chemistries have a larger deviation versus the idealized battery model. So this is for this PV plus lithium ion scenario, but you'll see the same observation or pattern for the other 2 scenarios. That's because the lithium iron phosphate or LFP battery has a flat discharge curve, which is consistent with the idealized battery model, which assumes a constant voltage. Meanwhile, the NCA and NMC chemistries have a steep voltage curve, which deviates from the idealized battery model. And as for the optimization, we found that in general, the optimum sizes predicted by the reduced order ECMs are larger than those predicted using the idealized battery model. Just to compensate for the larger sizes are required to compensate for any energy losses caused by the reduced order ECMs or predicted by the reduced order ECMs. Exceptions are this scenario here with the diesel generator, and this scenario with the grid connection, because in these 2 cases, it's more economical to just use diesel or grid instead of storing the excess energy. We also found that in general, the NMC chemistry has the largest sizes because it has the largest resistances. So you need more batteries to compensate for the energy losses. So to conclude, we found that the first order RC model is sufficient for modeling the lithium ion LFP, NCA, and NMC chemistries. The SOCs and optimum sizes between the reduced order ECM and the idealized battery model can differ by up to 5%. And so the idealized battery model is not applicable in scenarios with sharp demand peaks or if there's a cost competitive technology such as diesel or grid. In future work, we can apply this to other technologies such as lead-acid batteries, vanadium slow, and fuel cells. We can also integrate the degradation of lithium-ion batteries and experimentally verify the reduced order ECMs under pulse and discharge tests. Okay. So that's all. I would like to thank the ERDT scholarship as well as UP Diliman. Thank you.
Thank you, Michael. And indeed, very good energy modeling work from your end. Again, we'll reserve the questions later on as we proceed with our last but certainly not the least presenter. We have a science research specialist at the Research Center for Natural and Applied Sciences at the University of Santo Tomas. Okay, he'll be presenting on sustainable and environment-friendly energy storage device, banana cotton fabric with multi-walled carbon nanotubes and sulfonilic acid azochromotrope dope polypyrrole as supercapacitor electrode materials. So difficult to read. Huh? But anyway, Angelo, Nicole, Tolentino, the floor is yours.
Good afternoon, sir. May I share my screen? Sir, can you see my screen?
Yes, it's clear.
Okay.
Go ahead.
Okay.
Angelo.
Thank you, Dr. Kulaba, for the warm introduction. I would like to introduce myself. My name is Angelico Tolentino, and I'll be presenting to you sustainable and environment energy storage device, banana cotton fabric with multi-walled carbon nanotubes, and sulfonilic acid azochromotrope dope polypyrrole as supercapacitor materials. So for the introduction, we all know that 21st century is marked by significant changes in utilizing new sources of energy. One of these energy sources are the renewable energy sources as shown here. However, they have major disadvantages. One, their performance depends on the weather. Two, the energy release must be consumed immediately. And number three, they are non-dispatchable. So another energy source that we heavily rely on are the fossil fuels, particularly on coal. So according to the Department of Energy in the Philippines, more than 29 million metric tons of coal consumptions are used for power. However, they have major disadvantages. One, global warming due to CO2 emissions. Number two, increasing energy costs and consumption of fuels. And lastly, pollution. So to address these problems, we need energy storage systems such as the batteries, capacitors, and the topic for today, the supercapacitors. So what is a supercapacitor? Or the better question would be, what makes a supercapacitor super over the two energy devices? As you can see on the table on the left, supercapacitors have higher specific energy than a capacitor, which means it can carry more energy than a capacitor. Number two, it has a higher specific power than a battery, which means it can release tremendous power than a battery. And of course, they have a shorter charging and discharging time. It is easier to charge. And lastly, it has a longer lifespan. So to summarize these numbers, you will produce this plot, the Aragon plot of energy devices. And as you can see, supercapacitors bridge the gap between the batteries and the capacitors, and therefore they have a battery-like and a capacitor-like character. So for the main parts of the supercapacitor, we have the current collector. Number two, separators impregnated with an electrolyte, which provides ionic conductivity between the two electrodes and separates the two electrodes. And of course, the electrodes. So for the materials to be suitable supercapacitor electrodes, they must satisfy these characteristics. One, it must be porous. Two, it must have a high surface area. Number three, it must be conductive. And number four, it has the ability to store charges. So because of these characteristics, electrodes are considered the heart of a supercapacitor. So one of the materials that was used in this study is the banana cotton blended fabric from Philippine Textile Research Institute of the OST. It is made up of 30% banana and 70% cotton. They were used because they are highly porous. However, they are insulators. So to circumvent this problem, we use conducting and electroactive materials. One of the two are the multi-wall carbon nanotubes, or which is a carbon-based material. As you can see, it was used because of its high theoretical surface area, very high theoretical conductivity, high cyclic stability. However, they suffer with low specific capacitance. So to account for this low specific capacitance, we use conducting polymers such as polypyrrole. So they're used because they're easy to synthesize. They have a high conductivity. They have a high theoretical specific capacitance. However, they have low cyclic stability or degrade spuds. So you can see at this structure of the polypyrrole, there is a negatively charged ion that neutralizes the positive charge polypyrrole chain. And this anion is called the dopant. And the dopant affects the conductivity and the electrochemical performance of a supercapacitor. So that's why two dopants were used in this study, which is the chloride and the sulfonilic acid azochromotrope or SPANs. So for the chloride, It is a small anionic dopant. So since it is small, it is more mobile. And as a result, it can easily move in and out of the interstices of the polypyrrole. On the other hand, SPANS is a large planar polyaromatic polyanionic dopant. Since it is large, it is less mobile, and it is entangled within the interstices of polypyrrole. And as you can notice, there are 3 sulfonic groups that can dope several polypyrrole chains. And this effect is what we will be seeing later on. So the main objective of this study is to prepare and characterize banana cotton fabric, or the BCF, with polypyrrole, PPY, and the multi-walled carbon nanotubes as electrode materials for supercapacitors. Furthermore, to characterize these composites using SEM, thermogravimetric analysis, conductivity, cyclic voltammetry, or CV, and the galvanostatic charge discharge. And also as a proof of concept to prove if the best supercapacitor electrodes can light up a 3 volts light emitting diode. So for the methodology, for the preparation of the multi-wall carbon nanotubes dispersion, it is just a mixture of multi-wall carbon nanotubes, SDS, or sodium dodecyl sulfate, and water. And then it was the mixture rather was sonicated until well dispersed. And there you have it, a multi-wall carbon nanotube dispersion. So for the preparation of the chloride doped polypyrrole on fabric, or PPY ClBCF, basically the fabric was immersed in the pyrrole solution for an hour, and then ferric chloride was added dropwise into the pyrrole solution to initiate the reaction, and it was allowed to react for 2 hours under an ice bath. And then after that reaction, it was washed with ethanol and dried for 24 hours in a desiccator. And there you have it, a PPY ClBCF electrode. So for the SPANS doped polypyrrole on fabric, it's basically similar to the previous one, but the difference is that the fabric was immersed in a mixture of pyrrole SPANS solution. Ferric chloride was added dropwise. It was allowed to react for 3 hours under an ice bath, and afterwards it was washed with ethanol and was dried for 24 hours in a desiccator, and you have a PPY SPANS BCF electrode. So for the preparation of the binary BCF composites, the PPYCl multi-wall CNT BCF and PPY SPANS multi-wall CNT BCF, the fabric was first immersed in a MWCNT dispersion. It was dried and immersed in a deionized water for 30 minutes. It was dried again and placed in a desiccator, and the multi-wall carbon nanotubes will be deposited onto the BCF fabric. And then we apply the methods for the PPYCl BCF and the PPY SPANS BCF, and you have the 2 binary composites. So for the results for the thermogravimetric analysis, so using these thermograms, we can determine the degradation temperatures of all composites. So as you can see, all BCF composites showed good thermal stability. So for the conductivity values of the BCF composites, as you can see, PPY SPANS multi-wall carbon nanotube in BCF had the highest conductivity value of 0.41 Siemens per cm squared. This is basically a combination of the high conductivity of the multi-wall carbon nanotubes and the improved conductivity of polypyrrole due to SPANS, because SPANS can dope several polypyrrole chains. So for the scanning electron microscopy, as you can see, these small globular particles are the polypyrrole, and these are the major results. So using a chloride dope polypyrrole produces this diameter range. On the other hand, using SPANS as a dopant for polypyrrole, the diameter range increases. So therefore, it could be said that SPANS reduces the diameter of polypyrrole particles, and therefore it will increase its porosity. So this increased porosity will have a significant effect in the electrochemical results that I will be showing you in the following slides. So for the cyclic voltammetry, the picture on the left shows the cyclic voltammograms, and these voltammograms were used to calculate the areal capacitance of all the composites. And as you can see, PPY SPANS multi-wall carbon nanotube BCF had the highest CV areal capacitance of 785.45 millifarad per cm squared. So for the galvanostatic charge discharge, this is basically your sample. You will charge the sample until it reaches the peak voltage, and then you release the charge under constant current. That's why it is galvanostatic. So, and again, polypyrrole spans multi-wall BCF has the highest GCT aerial capacitance of 647.65 millifarad per squared centimeters. This is also due to the increased porosity. It is analogous to the sponge. Since the sponge is porous, it can hold more water molecules. The same thing here. The more porous the composite is, the more charged particles can hold onto it, thus having a high aerial capacitance. So using the binary composites to calculate for the energy density and the power density of polypyrrole multi-wall carbon nanotube BCF composites, the big question here is this really a supercapacitor? So calculations show that both devices are supercapacitors. And last but not the least, as a proof of concept that these electrodes can really store charge, we use the LED lighting test. In these connections, 2 AA batteries were used to charge the fabricated supercapacitor electrodes here. And then once it is fully charged, it was connected to the 3 volts LED and photodiode using a slide switch. And as a result, the LED lights up. So looking at the table on the right, comparing the 2 binary composites, both composites can be charged in just 30 seconds. And therefore, and again, polypyrrole spans multi-wall carbon nanotube in BCF had a longer LED lighting time of 161 seconds or 2.68 minutes. So for the conclusion, or to sum this up, polypyrrole spans multi-wall CNT in BCF composites had the highest conductivity, highest CV aerial capacitance, highest GCD aerial capacitance, high energy density, high power density, and also a longer lighting time. Because of these characteristics, it could be a promising material for supercapacitors. It could power small devices, and it could be used in rural areas, especially in off-grid areas. So for the recommendations, further GCT studies will be conducted, and supercapacitor electrodes can be improved using metal oxides, conductive polymers, and organic electrolytes. So these are my references. I would like to acknowledge the following people and organizations: Dr. Cristina Ramirez Peñafel, Dr. Cristina Binag, my scholarship from the DOST SEI, the fabrics from the PTRI DOST, the institution where I work on, the Research Center for Natural and Applied Sciences, and of course, the Graduate Student Fellowship provided by the Philippine American Association of Science and Engineering. Thank you, stay safe, and God bless.
Thank you, Angelico. That's also very interesting, study. Now ready for the question and answer portion for the last 3 speakers. Are there any questions from the floor, from our audience? Maybe, Amy, can you start the question from your end?
Okay, may I read some questions? First of all, these are excellent studies. Congratulations to the presenters, and thank you for showing us these promising technologies. Let me ask a question for Juvenile. What are the future plans in this study?
Actually, I'm finalizing my methodology on my plan to study on the molecular dynamics and mathematical modeling of simultaneous mass transfer and transesterification reaction on the in situ production of fatty acid ethyl esters from this mahogany seed. And this is actually my dissertation on a PhD.
Thank you, Engineer Cartel. Amy, you still have a question there?
Yes, another one for Angelico. Why did you use multi-walled carbon nanotubes and not other types of carbon?
Hello. Can you hear me? Okay.
Yes.
I can hear you.
Actually, multi-wall carbon I used multi-wall carbon nanotubes because it has a high conductivity basically, and also when you deposit it into the banana cotton fabric, the structure of the multiple carbon nanotubes becomes more porous. So because of that, more ions can enter into the spaces of the the electrode composite, and thus you may have a high specific capacitance.
Thank you, Angelico. Cynthia, from your end.
Sure. This is for both Michael and Angelico, just a question of the economic and I guess policy implications of your findings.
Ma'am, come again po.
Can you expound more on the economic implications as well as potentially social and policy implications of your findings?
Thank you, Madam. And also thank you for that question. Basically, for the economic implications, of course, we all know that some energy storage devices use some chemicals that are very dangerous to the health and also to the environment. And also for these effects, we can decrease the use of these dangerous chemicals that can cause explosions like that, because the supercapacitors can what would I how would I put this, it doesn't generate overheating, I may say, because multiple carbon nanotubes, for example, doesn't involve electrochemical reactions. That's why it cannot produce more heat. That's, I think, I hope I could I did answer the question well.
All right. So for my study, the economic implications that you need more batteries to power off-grid energy systems. So yeah, that's the main economic implication. So that will actually increase the costs of the system slightly.
Okay. Any more questions from the audience?
Sir?
Yeah, go ahead. Who is that again?
This is Mr. Angelico po.
Yeah, go ahead, Angelico.
I think I have a serious question on Mr. Michael Castro. It's just a curious question. Is there a possibility that a battery and the supercapacitor can be combined in a single device? Just a curious question.
Okay, I'll try to answer that. In a power system kasi, yung supercapacitor, that has a role when you're trying to process high frequency signals. So I guess it's also possible to use them together in a energy system. Now, as for the same device, I can't give any specific example, but what I can tell you is that it's possible to use them together.
Okay. Thank you.
Okay. Any more questions? It's actually 11:54, close to noontime and almost midnight, in the East Coast. So we might have to wrap up this session. Again, thank you so much to Dr. Redillas and Ray Frias, to Cheryl Mariano, Anne Elizabeth Rolion Innova, Jovenil Cartel, Michael Castro, and Angelico Tolentino. You have excellent, studies, and I hope you can share further, the results of your work as you, continue your research. And I would like to request at this point for everyone to have a photo op. Cynthia, can we do that? Can we request everyone to open your cameras if it's possible? And then Cynthia and Amy, can you take the photos, please?
Yes, sir.
Oh, how do we do this?
I think there are two
panels. Yeah. So
Okay,
there would be two. I will count to three, and you guys can smile. All right. One. Oh. Okay. All right. Okay. 1, 2, 3. Great. Okay, one more. Let me save that. Save. Right, sorry. Do another. 1, 2, 3. Great.
Okay.
Nice.
So again, thank you so much for participating in this session. If you are interested to join, by the way, this rec, please do contact PAASE. Would gladly want to engage you in more discussions with your work. And I hope, in the future, you can join, the Academy. So again, thank you so much to the excellent speakers that we have. Continue with your good work. Okay, and to my assistants here, Cynthia and Amy, thank you so much for your assistance and for everyone here who participated in. Stay safe, everyone. Keep safe and healthy, and of course, be happy. Okay. Again, thank you so much, guys. You still have other sessions tonight, the Philippines, morning in the US, and on Monday, tomorrow. So we still have a full schedule for the APAMS 2021. Thank you so much. Enjoy your lunch and good night for those in the eastern part of it. Thank you, and
Yes, sir.
Thank you, everyone.
Thank you.
Everyone.
thank
Thank
you.
you.
Thank
Good
you, everyone.
afternoon and good